Hi, I'm Nicholas Fernandez. I'm a computational scientist at the Human Immune Monitoring Center at the Icahn School of Medicine at Mount Sinai. And this video is going to walk you through a short example using the Jupyter widget Clustergrammer2 to explore a bulk gene expression dataset called the Cancer Cell Line Encyclopedia, produced by the Broad Institute. So we're going to start up with the Clustergrammer2-notebook GitHub repository. And if you scroll down through the README, you can see we have links to several examples. For this video, we're going to go ahead and launch a Jupyter Lab instance using MyBinder. So we open this in this tab, and this uses a service called MyBinder to launch a live Jupyter notebook for you to actually work with. So this can take anywhere between 30 seconds to a few minutes to actually launch. And okay, so we now have our Jupyter Lab instance running, and we have you can see the launcher here and the sidebar. So if we go into double-click notebooks in the sidebar and click 2.0 CCLE gene expression, this takes us to our notebook. So the Jupyter Notebook, just in general, allows you to combine text, code, and visualization. So this is an editable document, and you can execute it. So we're going to run the first couple lines of the notebook, and we're importing Clustergrammer2, and we're loading our data, which is we're loading our data into a pandas data frame. So our data set is contains 18,000 rows or genes and 1,037 cell lines or columns. So we're going to use Clustergrammer2 to visualize this data set. And we're going to only visualize the top 1,000 genes based on variants. And we're going to generate two views of that. So the first view, we're just going to look at the raw gene expression data. And so Clustergrammer is now filtering the data and performing hierarchical clustering on both the rows and columns, and then generating a widget to visualize the data. So now we're able to visualize our data set. So we have 1,037 columns, so shown here, and we've looking at the top 1,000 variable genes. And if you hover over the rows or the genes, so you can zoom in and see specific expression level of a gene in a cell line, and we've added several different categories to our cell line, so we can see which tissue cell line is from, and we can see that cell lines tend to cluster based on their tissue of origin, and you can see other patterns in your data, so you can reorder your cell lines by tissue and see different expression patterns. You can zoom in and start to see which genes are differentially expressed. And then below it, we're using the normalize method to perform z-score normalization on the same data set as we saw before. So now we have z-score the genes across all our cell lines. So now this helps us visualize a bit better the differential expression. So now we can go ahead and pick a particular cell if we want, and reorder the expression of all the genes based on that particular cell type. And then we can start to observe patterns in our data and understand which cell lines are clustering near each other, what gene expression patterns are involved in this, and just really like interactively explore the data set. So here we've reordered our cell lines based on overall expression, so we can see some cell lines just tend to express certain genes higher than others, and if you reorder based on the expression of hematopoietic and lymphoid tissue, which appear to be a sort of outlier cell line, you can see this nice gradient that occurs of these genes in one particular cell line, and sort of shifting different tissues. Gene expression is shifting across different tissues. So Clustergrammer allows you to really dive into a complicated high-dimensional data set, and here we're very easily able to explore this over a million data point data set using Clustergrammer. So we hope you enjoy the notebook, and thank you.